All right, I guess it's time to get started on our RNA Collaborative seminar of today. In fact, you get 4 seminars for one. It's my particular pleasure to kick off today's seminar series that is hosted jointly by the University of Michigan Center for RNA Biomedicine and the Regensburg Center for Biochemistry. And today we have two speakers from the University of Michigan. First one will be Julian Zhu, who received his undergrad bachelor's in biotechnology from Nankai University, one of the top universities, research universities at China, and then got his Ph.D. in medical sciences, physiology, and pharmacology at the University of Florida, working with Weihong Tang. Then did a postdoc at NIH or NIBIP in Bethesda with Jiajun Sean Chen, to then start his assistant professor position at the Department of Pharmaceutics in the School of Pharmacy in Virginia Commonwealth University, which is in Richmond, Virginia. And then in 2023, we recruited him here. The School of Pharmacy recruited him as the Garo Paul Endowed Associate Professor in the Department of Pharmaceutical Sciences, College of Pharmacy, where he now also serves as an associate chair for the department. He has lots of awards in his name, including the Young Investigator Award from Biomaterials or the Mid-Career Investigator Award from the Chinese Association for Biomaterials, and really bridges the RNA material science fields in the pursuit of RNA medicines. Today, he will be talking about engineering and delivery of circular RNAs, therapies, and vaccines. Julian?
All right. Thank you for the kind introduction, and thank you everyone for attending. Niels, I hope I can live up to your introduction. Hi everyone. I go by Julian. My name is Guizhi Zhu. I'm going to share some of the research in our group related to circRNA engineering and delivery. This will be a high-level talk, but I hope that I can give you a idea of what we work on in the lab. So overall, we work on the nucleic acids, surrounding nucleic acid. We work on the engineering, delivery. Those are some of the representative projects in the lab. Those highlighted are going to be touched upon today and very superficially using engineering approach to develop synthetic circRNA as vaccines for cancer, infection disease, or as therapeutics for protein or RNA replacement therapy in in genetic disease, or develop a sort of a specialized delivery systems for many of those RNA therapeutics, including circRNA, mRNA, and in some cases, oligonucleotides. I wanted to start by two properties that many of you are aware of and were also intrigued by for circRNA. One is the stability, and the second, I think, is related, is the relatively low innate immunogenicity. So I wanted to show one stability study, which let me have a laser point, in which we used a circRNA that is embedded with a broccoli aptamer. Many of you know this. This broccoli aptamer are fluorogenic only when they form a correct formation in the presence of a substrate small molecule, and we use this to study the biostability so that it can help us to tell or distinguish linear RNA and circRNA from circRNA. So we compare the linear broccoli and the circular broccoli, and we transfect those two RNA into a live dendritic cell, and we monitor the fluorescence intensity by flow cytometry at a several time point post-transfection. On the left column, what you see is the quick fade away of fluorescence intensity of those cells after transfection for a linear broccoli aptamer. On the right, you will see that this fluorescence signal will stay there for quite a bit of time. A quantification shows that they can stay for at least about 7 days. In this case, this is a semi-quantification, and this doesn't offset the signal dilution caused by cell division. But this demonstrates the stability of the high stability of circRNA relative to linear RNA. What this entails for a coding RNA is, again, many of you know that the linear RNA relative to linear RNA, the circRNA can have a prolonged translation duration. This can be particularly true for soluble protein. May not be that much different for a lot of transmembrane proteins or otherwise very stable proteins. But for soluble proteins that are themselves have a relatively short half-life, then the translation kinetics for RNA can become very significant. And I think related to that is that the unmodified circRNA can have a relatively low pro-inflammatory response. This is a comparison of a unmodified circRNA in comparison to a modified 5-MU modified mRNA. So based on that, we are interested to explore some of the application as well. One of the first application we study is to use those small circRNA that are synthesized from RNA oligo using DNA splint as a excipient. And upon ligation, they will form a circle, and then we can purify this RNA to remove the DNA splint or the unligated linear RNA. In the circRNA, we have a RES and a COSAC that help to drive translation. We also have a third region, which is which can be customized to express the peptide or proteins. For small circRNA, we're interested to express peptide antigens for diseases that, for which the cells that we wanted to kill. One notable example is cancer cells. Now, we can design the circRNA to encode one or multiple peptides or MHC class 1 or class 2 antigen peptides, depending on which type of T cell, CD4 or CD8 T cell we wanted to elicit T cell response against. And we often use the lipid nanoparticle to deliver this circRNA. In this case, we use the lipid nanoparticle to deliver circRNA to draining lymph nodes. Within the draining lymph node, they will be taken up by antigen presenting cell primarily and then present antigen to elicit T cell response. Because they are stable, they can prolong the translation. This also benefit the T cell response potency and duration. One of the T cell response study we did is in mice. We immunized the mice with those vaccine in comparison with benchmark 3 types of a benchmark mRNA, modified mRNA vaccine, and monitor their peripheral blood T cell response against this synthetic peptide antigen. At a very low dose, 1 microgram, you can also already see a very high T cell response, high relative to this dose. If we increase the dose, you will see increasing dose-dependent increase of a T cell response until it's saturated at about 30 microgram. And if we stop the dosing, the T cells are going to die off and fade away. But the T cell elicited by circRNA is the most long-lasting. And this makes them very interesting for a lot of disease therapy, including tumor therapy. This one example of many is the SM1 BRAF V600E mutation carrying melanoma. This tumor didn't respond to even a dual immune checkpoint inhibitors, which is the most potent immune checkpoint inhibitor therapy in the clinical for melanoma. But if we combine this with a circRNA that now encode 4 peptide antigens, 4 out of the 8 tumors were actually completely regressed without recurrence. Encouraged by this, we'll also explore the further application for small circRNA. One of the applications is guideline, circRNA guideline. We explored their application in ADAR-based editing or K13D application. So in the ADAR editing, in the case of ADAR editing, we can achieve actually up to 70% of editing efficiency for this target. But I'm not going to discuss too much detail, so I can have some time to discuss the delivery side of this type of RNA. So in the delivery side, I will mainly talk about two closely related delivery system that both incorporate a material, FDA-approved material called polymer surfactant. We incorporate them in two slightly different ways for pulmonary delivery, deliver the RNA to the lung, and for two different applications in either mucosal immunity vaccine or in genetic disease or specific cystic fibrosis. So I wanted to quickly go over how we make this particle. So basically, incorporate a polymer surfactant by either thoroughly mixing them, or by coating the nanoparticle with this polymer surfactant. This polymer surfactant is an FDA-approved product used in preterm baby by intratracheal application for decades. So that is a pretty safe excipient for this type of materials. In the case of a mucosal vaccine, many of you may be aware that during the pandemic, many tried to give the mRNA vaccine into the lung, but unfortunately, they didn't elicit significant mucosal immunity. So after several years of research, we found that if we have a hybrid immunization by intramuscular priming followed by intratracheal or intranasal booster, and within the booster, if we deliver a circRNA and by this polymer surfactant incorporated lipid nanoparticle, it will elicit actually pretty potent IgA response and non-resident T cell response. And specifically, we found that they can this type of lipid nanoparticle can now efficiently deliver the RNA to alveolar macrophage, which is a central antigen presenting cells, to elicit mucosal immunity and promote TGF-beta production, which is central to the mucosal immunity modulation. We then explore their application for SARS-CoV-2 protection and lung metastatic cancer therapy. This figure shows the comparison of a lipid nanoparticle versus polymer surfactant lipid nanoparticle or PS lipid nanoparticle for RNA distribution. So PS nanoparticle promote the retention of the RNA in the lung and reduce their dissemination to systemically or to the liver. The IMS data, which shows the RNA expression, confirm this result. In a single cell level, we use a dye-labeled RNA that also express AppLook, and in biopsy level, we can examine the dye fluorescence intensity of the RNA in a specific single cell level. We again confirm that the polymer surfactant nanoparticle can increase the alveolar macrophage uptake of the RNA. This was further confirmed by using an AI14 reporter mice that deliver a mRNA Cre recombinase that again confirmed the efficient delivery of polymer surfactant nanoparticle in alveolar macrophage. Then in mice, you see after priming booster with those circRNA delivered by polymer vaccine, they do elicit slightly higher mucosal immunity than even the mRNA delivered by the same polymer surfactant lipid nanoparticle. This is shown by both the IgA humoral immunity and the T cell response, especially non-resident memory T cell. So this motivated us to test their ability to resist the viral challenge in collaboration with Dr. Sang at the University of Virginia. And what we did is that we again immunized the mice by hybrid immunization like this. And then 90 days after priming, we challenged the mice with SARS-CoV-2 Omicron. And we found that in both as well as the nasal wash, the hybrid immunization in which the booster is delivered using a lipid nanoparticle coated with a pulmonary surfactant for a circRNA vaccine, resulting in the lowest viral titers in both mouth and the nasal wash. Encouraged by the T cell response, we are also interested to explore their application in non-resident tumor cell therapy. So in this study, we established a pre-existing lung metastatic melanoma for B16F10 murine melanoma. And then after 7 days of tumor injection inoculation, we start to immunize the mice and also treat them together with the anti-PD-1 immune checkpoint inhibitor. And again, we show that the pulmonary surfactant delivered RNA exhibit the most potent therapeutic efficacy. At day 90, we changed all the surviving mice, and 6 out of the 7 mice survived after the challenge. In contrast to at most 1 out of 7 for the other groups. Another case of application is using those pulmonary surfactant coating nanoparticle to deliver this large circRNA encoding a CFTR transmembrane protein. I'm going to hurry up because of the time. In this case, we actually wanted to deliver those to epithelial cell, especially lung epithelial cells. I'm not going to play the video, but as you can see here are the lung bronchial epithelium that are all lighted up by the RNA carrier delivered by this pulmonary surfactant. This is again confirmed by using AI14 reporter mice at a single cell level that a PS nanoparticle promote the delivery. I will show just one last data in which we use a rabbit model of cystic fibrosis, and after a 1 dose intranasal administration, using a nasal potential difference assay, we can see that the CFTR function in the nostril of those rabbit can be recovered for up to 7 days, which sounds pretty encouraging for us. So with that, I hope that I showed you mostly some of the examples of synthetic circRNA engineering and delivery, and happy to discuss some details. And finally, I want to thank my group, collaborator, and funding source, and thank you all for your attention.
Thanks, Julian. Questions, please put them into Q&A. So here's one question by Madeleine Mayday. Thanks for your talk. For the melanoma study, 4 out of 8 mice had a complete response, but the other 4 did not. Why do you think that is? Was there an issue with circRNA delivery uptake, etc., or was there a biological reason?
Yeah, that is a very active research that we're trying to understand the mechanism of the resistance. One observation we had so far is that the tumors are controlled during the treatment. Once the treatment stops, then they quickly rebound. So I think the impression we got now is that for whatever reason, we have to eradicate them better during the treatment. Otherwise, the chance to eradicate the tumors is going to fade away once we stop the treatment. But we don't know exactly the reason for those tumors to resist the therapy.
All right. Thanks, Julian. In the interest of time, let's move ahead. I'm sure there will be more questions of this wonderful talk. And please put them in the Q&A, and Julian can ask them online, answer them.
Absolutely. Yeah, thank you.
All right, thanks, Julian. So next up is Sarah Kargbo-Hill, who got her undergrad degree at Penn State University in biochemistry and molecular biology, and then did her Ph.D. at Yale University with Danielle Colon-Ramos in cell biology, and then did a postdoc with the famous Jennifer Lippincott-Schwartz at NIH, together with and co-advised by Michael Ward. And then in 2024, started here in the Molecular, Cellular, and Developmental Biology Department at the University of Michigan. She also is a biological sciences scholar and a K99R00 recipient, as well as other awards. And so without further ado, she will talk to us about TDP-43.
Great. Thanks, Neil, for the introduction. I'm excited to share our work today with you on how TDP-43 regulated cryptic splicing is influencing RNA dysregulation in ALS and FTD. So in the lab, we're interested in ALS and FTD, which are a spectrum of disorders. These are two neurodegenerative disorders, a motor neuron disease, ALS, and a dementia, frontal temporal dementia, that share pathological features. So one of those features is dysfunction in this protein TDP-43. So TDP-43 forms these cytosolic aggregates in patients, which we can see here in these arrows. And so this is, a defining feature of the pathology. About 97% of patients with ALS will have these aggregates, as well as about half of patients with frontal temporal dementia. But one thing to notice is, in addition to forming these aggregates, there's also loss of this protein from the nucleus as well. And so these two features go together, and it's become apparent in the field that the loss of function may actually be the earlier preceding events. And so that is the one where we're very interested in focusing on. So TDP-43 is an RNA binding protein that has a number of different functions related to RNA metabolism. So these include things like RNA splicing, RNA transport, translation, RNA stability, and promoter silencing, to name some of the described functions. We're particularly interested in its role in RNA splicing, which is one of the better described direct roles for this protein. And so what TDP-43 does to regulate splicing is it acts as a repressing factor. So when TDP-43 is present, it will bind to regions of introns that we call cryptic exons in the field that will allow for repression so that these don't get spliced to the mature RNA. But when TDP-43 is not present, many of these now get spliced in. They're called cryptic exons in this field, particularly because they're not seen in sort of control or healthy samples, only upon loss of this essential protein. In any case, when you have the inclusion of the cryptic exons, some of them will contain premature termination codons or frame shifts that ultimately lead to loss of the RNA through nonsense-mediated decay, and then loss of the corresponding protein. So a few years back now, and during my postdoc, we had used human iPSC-derived neurons in order to understand TDP-43 loss of function. So it was important to use a human neuron model, as these splicing events are species and cell type specific. So using this system, we identified hundreds of these different splicing events that were enriched when we knocked down TDP-43. Around the same time, another group had used ALS, FTD neocortexes and had used FACS sorting in order to separate out neurons from these brains. And they were also able to identify very similar cryptic splicing events. And so in summary, really, there's hundreds of these events that you can find in neurons when you knock down TDP-43. And so we really wondered what these particular splicing events, what consequence they may be having on the cell. TDP-43 is an essential gene really required for cell viability and, affects hundreds of targets. And so we wondered if, loss of this splicing regulation may then be influencing some of the other changes that can be seen upon loss of TDP-43. Basically, is there sort of this cascading effect of these changes occurring? And so we used the human iPSC-derived neurons, and we again knocked down TDP-43. I'm just showing you that immunostaining here. And what we really wanted to do was to gain a systems-level view, really, of what's happening, right, for these hundreds of changes. And so we did RNA sequencing, proteomics, and a TDP-43 protein interactome, and generated all these data sets in order to integrate them together. And so this work was done in collaboration with Ernest Frankel's group at MIT and a really talented graduate student in his lab, Valina. And they use this omics integrator tool in order to take our data sets in order to build multi-omics analyses and protein-protein interaction networks that we could then use to generate and test hypotheses. And so this work is now out on bioRxiv, so I have that listed here. So if you're interested, take a look. And we also have a version of this network that's a web browser that you can use to actually see all the labels of these individual genes. And so there's a QR code here. But essentially, this is what our network looks like. Each of these different shapes represents a different gene protein node, and they're grouped in these different neighborhoods of sort of related GO-term functionalities. And so those are sort of listed here. The color of the shape is related to the proteomics score. So when we do proteomics, if the protein is increased upon knockdown of TDP-43, it's in red, and for things that are lost, it's in blue. And then green would just be anything that we had no proteomics information on. So it just no information. And then we also have these borders to represent genes that are differentially spliced upon loss of TDP-43. So what I want you to appreciate here is that these differentially spliced genes really exist across a number of these different networks. It's not really clustered around one pathway, but really quite dispersed for these sort of important pathways that are related to TDP-43. Also, and so, taking a look at these 13 neighborhoods, we've sort of categorized them into these even broader groups. And so over here, we have things related to nuclear function. We also have more synaptic or neuronal outgrowth neighborhoods over here, autophagy down here, and then different aspects of metabolism as well. So all of these are sort of important pathways that we believe are linked to TDP-43 function. So since this is an RNA meeting, I particularly wanted to highlight some of the RNA node here for RNA processing that's dysregulated. At the center of this here, we see TDP-43. The gene is actually called TARDBP, so excuse the nomenclature. But, of course, we've knocked it down, so we see really strong reduction here. And then we also see a number of other RNA binding proteins as well as splicing factors that appear upregulated, potentially in response to this. And in the I don't have time to get into it today, but in the bioRxiv, we discussed some of the other secondary splicing changes we believe may be potentially driven by some of these other RNA binding proteins activity as well. Okay, so essentially, though, from the systems level view, one of the goals we really had was to understand which of these different cryptic splicing events may be sort of the most impactful, and sort of how are they impacting the cell. And so, for our multi-omics analysis, one of the things we did was we characterized each of these proteins based on whether or not it also had a splicing change or an RNA level change. And so looking at this flow chart, we can see that for genes that have differential, that were measured in the proteomics experiment, most of them are not misspliced, right? But some of them are. And of those, some of those are then differentially, measured at the RNA level. And then a smaller subset then here is differentially expressed at the protein level. And so we can kind of look back at these protein level changes and ask sort of maybe what caused it, right? Or, what it could be associated with. And what we found, one of the major findings we had from this analysis was that for differentially expressed proteins, this group here that were also differentially spliced, they actually had the largest effect size changes in protein levels. So these are really our potential drivers of other changes. They're really the big effectors here. And so that's shown here in this plot where we can see change in RNA and change in protein levels. We've also highlighted in blue those that actually contain those cryptic exons. And it's a really high number of these that have, those types of events that really only appear upon loss of TDP-43. And so we wondered if any of these cryptic splicing events may be important for neuron survival. One of our goals is to understand, which of these splicing events could be important for disease, potentially to target it. And so we wondered if any of them occurred in other essential genes. And so we thought this may be possible also because when you knock down TDP-43 in our system, we're able to see a decrease in neuronal fitness over time. So these are longitudinal imaging experiments where we capture images of neurons over time, and we can kind of quantify that here. And so in order to understand all the other genes in the system, we conducted a CRISPR survival screen where we knocked down every gene in the human genome, and we measured frequency over time. And when we do this, we can identify about 2,000 genes that are essential for neuron survival. These are in many pathways related to ribosome biogenesis, protein homeostasis, and many others that I'm not going to show today. But essentially, we now have a list of genes that are essential, and we can take a look and compare them to those genes that we find are differentially spliced. And so that's what we did. So we took that list of genes that are differentially spliced that also will lead to reduction of the gene. And so that's this group here. And then we plotted their survival phenotypes. And so what I hope you can appreciate is that there really aren't that many that are essential, but these two here that I've highlighted in red really caught our attention, MNAT1 and CDK7. And these two essential genes were really interesting to us because they belong to the same protein complex. This transcription factor 2H complex that's part of the general transcription machinery that's important for interacting with RNA polymerase in transcription. And so here I'm showing you the plots of these splicing events to show you the cryptic exons. So, here we have an exon, and then this would be the intronic region. And then when we knock down TDP43, we can see that we get some reads now in this region where, there's we call this the cryptic exon because it's not present in the control sample. We see the same thing in MNAT1 for this region here. Both of these cryptic exons lead to premature termination codons, and we've measured them and found reduced levels of total RNA and protein as a result of these cryptic splicing events. And so we wondered what effect loss of TF2H may be having on potential RNA within the cell, in particular in these neurons. And so in order to address that question, we used a nascent RNA imaging strategy. So in this method, we pulse cells with EU, which is an RNA nucleotide analog, for 30 minutes. And then we use click chemistry in order to visualize the RNA within the cell. And so this would be, total RNA. And when we do this, we can see that, we're able to see the nucleus within these neurons here that I'm showing you. We also have here stained TDP43 just as another sort of control to follow. So you can see these are the control cells. Now, when we knock down TDP43, we can see that reduction, and we also see reduction in the global production of RNA during this period. If we then add back antisense oligonucleotides, or ASOs, we actually are able to rescue this nascent RNA. So these ASOs are these small oligos that we've designed to direct to the CDK7 and MNAT1 cryptic exon regions. So they essentially act like TDP43 by sterically blocking these cryptic splicing events, allowing restoration of normal levels of CDK7 and MNAT1. And so using this tool, we're now able to rescue these levels, suggesting that this global reduction in this RNA, nascent RNA production is due to those cryptic splicing events. And so here's this quantification here, where we can see the control sample, the TDP43 loss, and then this rescue. Importantly, only when we rescue both with the CDK7 and MNAT1 ASOs are we actually able to see the strongest rescue. So today I told you a bit about how we can use multi-omic strategies in order to understand some of the systems level changes that are occurring upon TDP43 loss, that we can use an integrative network strategy in order to understand the pathways that are related to TDP43 function, some of them highlighted again here, including synaptic biology, nuclear biology, autophagy, and metabolism. And then ultimately, one of the directions that we found in the lab is that cryptic splicing events, particularly in that CDK7 and MNAT1 genes, are actually sufficient to regulate nascent RNA levels within the cell. And so with that, I would just like to thank my group, who many of whom have contributed to this story, as well as our collaborators, Ernest Frankel and Velina Kochevera at MIT, and many others. Like I had mentioned, the multi-omic story is now on bioRxiv, and we have the QR code here if you're interested. Thank you for your attention, and happy to take any questions.
Great. Thanks, Sarah. There is one question that we can handle here. Are there splicing events from Bikash Saho? Are there splicing events associated with TDP43 abnormalities like mutation and mislocalization that are disease associated? Could you please comment on their impact on C90RF72 splicing events?
Yeah, sure. So certainly if TDP43 is really heavily regulated, the level matters a lot. And so there's actually different types of missplicing events that will occur if you overexpress TDP43. It's a lot more exon skipping events people have found as well. And so certainly the levels matter. I haven't seen anyone who sort of attributed the pathogenic forms of TDP43 specifically to differential splicing, I think typically it's through loss of function of those mutations. You can see some of the same types of events as if you would knock down the gene. Yeah. And so in C9orf72, you see some of these same, actually in that sorted patient neurons, I believe some of those are C9orf72 carriers. And so that will lead to some of the loss of function as well.
Great. Thanks, Sarah. And in the interest of time, I'll hand over to Regensburg.
Thank you so much, Niels. So welcome, everyone. I'm Neva Çalışkan, heading the Department of Biochemistry team at the University of Regensburg. And today we have two exciting talks on the Argonaute. So let me first introduce Professor Gunter Meister. His talk will be on navigating Argonaute proteins through gene silencing cycles. Gunter Meister is a professor and chair for Biochemistry 1 at the University of Regensburg. He received his master's in biology from the University of Bayreuth in Germany. Then he moved to Max Planck Institute for Biochemistry in Martinsried. And after this, he was a postdoctoral fellow at the Rockefeller University in New York. And in 2005, he started his own lab at the Max Planck Institute for Biochemistry. And since 2009, he is a full professor and the chair of biochemistry. So please, let's welcome Gunter, and you could share a screen. Thank you, Gunter.
All right, thank you very much, Neva, for introducing me and the opportunity to talk a little bit about Argonaute proteins today in this seminar series, which is our topic here at the University of Regensburg. So Argonaute proteins are really at the heart of microRNA-guided gene silencing. They bind to a microRNA, and the microRNA guides these Argonaute proteins to 3' UTRs on mRNAs. And then when they find their target sites, Argonaute proteins actually recruit a member of the TNRC6 protein family. These are really remarkable proteins, which are unstructured scaffold proteins that now form all the downstream interactions. They interact with the poly-A tail, with the deadenylase complexes, leading to deadenylation, recruitment of the decapping enzymes, leading finally to decapping and 5' 3' exoribonucleolytic decay. So these nanomachines are not only made to recruit these scaffold proteins, but since they are really at the heart of microRNA-guided gene silencing, they need to be generated first, they need to find their target site, and they need to be tightly regulated. And this is what we're working on, and it's actually far more complex as one would think. So the current view is that Argonaute is made and then folded using classical heat shock proteins such as HSP70. Then they need to receive their microRNA load, and microRNAs are made by DISA. DISA enzymes bind the microRNA precursor and process a short double-stranded RNA, which is then loaded into an Argonaute protein, and only one strand is loaded. Whether DISA is the only important enzyme for loading, or whether there are other proteins like heat shock protein 90 are important for this, is still a matter of debate, at least in humans. Then Argonaute proteins are guided to mRNAs. They recruit TNRC6, and then at some point when the mRNA is degraded or not degraded, just repressed translationally, TNRC6 proteins have to be released, and Argonaute protein is now free again for maybe a new round of gene silencing. We have shown, and I would like to tell you a little bit about loading at the beginning here, and then a little bit more about the regulation of these nanomachines. So we have shown in work together with Dina Grohmann and Sarah Wilcom a couple of years ago using single molecule FRET experiments that Argonaute proteins actually travel through conformational dynamics and through different conformations. And this is some examples here. I do not need to go too deep into the method here. So there's FRET efficiency with 2 fluorophores on Argonaute at very specific positions. And the efficiency, the higher the efficiency is, the closer the fluorophores are together, and the lower they're further, the far away they are. You can see this is the APO enzyme, Argonaute free. This is a microRNA bound Argonaute, and that is a microRNA bound Argonaute and a target RNA. And the only thing that I would like to draw your attention to is that when we look at dynamic molecules from the APO state to this ternary complex with the 3 components, we see that it's getting less and less flexible, and Argo moves into a more closed conformational state when it's bound to a target. Having said that, when the target is released, Argonaute needs to also go back into a more open state to maybe receive a new RNA again. And this is what we are trying to work on and to figure out. So coming back to DICER and really Argonaute loading with a small RNA, very early cryo-EM data found already particles where not only DICER was present, but also another density. And in these very early negative stain particles here, people were able to model Argonaute in there. And we and others a long time ago had shown that this is actually a complex of Argonaute and DICER. And we found that DICER now actively really engages in loading of the small RNA into Argonaute proteins, which we call risk loading. So that is actually happening at a very specific step during the cycle of gene silencing. So DICERs define the small RNA product and also directly load them onto Argonaute proteins. And there's a little bit of ongoing work here towards risk loading. This is a very exciting project together with Richard Stefel, Peter Svoboda, Dina Krohmann, and Stefan Ameres, where we now look into this loading process in a little bit more detail. And when we now look at DICER and go away from the classical view where we have these domains here present, the folded domains, but look at the entire sequence of DICER, we found that 30% of human DICER is actually unstructured. So they're actually mainly producing intrinsically disordered regions, unstructured loops, and that's about 30%. And people have not yet investigated this 30% of DICER. And since the Argonaute can actually be found here, this density, we thought, okay, these loops, and especially this green loop here, IDR3, could be important for binding of DICER to Argonaute. And this is what we tested here. And this is really ongoing work. I'm just jumping into some of our biochemical work here. So we use this green loop in isolation, shown here. On the other half, we pull down this isolated loop here and see whether it actually also brings down Argonaute. And indeed, the green loop nicely binds to Argonaute. There's a little bit of background here. If we turn this around, we pull down Argonaute, you can see that this green loop can also be pulled down. So this green loop really binds here. When we look at wild-type DICER and a DICER variant that lacks this green loop and do the binding experiments, again, you can see here that DICER does not bind to Argonaute 2 anymore when this green loop is removed. So actually, this intrinsically disordered region is required for Argonaute binding. Is it also required for Argonaute loading? And here Lisa Betta in the lab developed an assay where we can biochemically really address that. It goes back to an assay that I developed when I was a postdoc really long time ago. And this is shown here. We overexpress DICER, MYC DICER in this case, and flaggage ATAC Argo2 in cells, and then generate lysate. We IP Argonaute 2 here, and that brings down the bound DICER. Now we can use a microRNA precursor, in vitro transcribed synthetic microRNA precursor that is unrelated, not present in the cell. Here we use EBV microRNA, added to these beads. Now DICER produces a small RNA, and if it loads it properly into Argonaute, we can actually monitor Argonaute activity later on by simply adding a substrate RNA that is radiolabeled and can monitor cleavage of the loaded RNA. So that now confirms DICER binding, processing, and active loading. And this is shown here. It really works well. You can see that we can have two substrates, one for the 5p strand, so both of the strands can be loaded, and one for the 3p strand. You see this is asymmetric here. The 3p strand is better loaded than the 5p strand, which is consistent with the sequencing results as well. And now when we look at the delta green variant, when we overexpress this DICER variant, you see that we have much less cleavage activity, confirming that these intrinsically disordered regions recruit and bind Argonaute proteins. And as a side note, these IDRs are actually charged. And our current working model is now that these charged IDRs mimic small RNAs during the loading process. So they occupy with the charges the binding groove of the microRNA and are subsequently replaced by the microRNA during loading. Now, a few more words on regulation. So this is now loading and generation of this nanomachine here. Now, how is that regulated? And that would like to tell you a little bit about our phosphorylation work. And that is now really a couple of years ago where we asked are Argonaute proteins actually modified post-translationally and looked at Argonaute phosphorylation. And we found using mass spectrometry, when the isolated human Argonaute 2, we found peptides that are heavily modified. And we found peptides that even carry 4 phosphates at the same time. So this is really a hyperphosphorylated peptide. And that these peptides derived from the C-terminal end here of Argonaute 2 in this case, and this is also conserved throughout actually all Argonaute proteins that deal with microRNAs. And this is a phosphorylation cluster that can carry up to 4 phosphates at the same time. We characterized this a little bit further, but that has all been published years back, and we found that when we use phosphomimicking mutants and look at mRNA binding, so not looking at microRNA binding, but mRNA target binding, we see that when the phosphates are there, the negative charges are there, the mRNA can no longer be bound. We can do this also on a more global scale. We lose mRNA binding when we use glutamate mutations at these sites. And I took this from a paper from Joshua Tor that was published also a couple of years ago. This is actually the loop where these phosphates sit. And you can now see when there's phosphates on these serines, these phosphates clash with the phosphate backbone of the mRNA and simply replace the mRNA and dissociate Argonaute from the substrate. So they are located on a loop within the PV domain, and it reaches into the mRNA binding channel, and the mRNA is released. To sum this up, because it's all published here, and we contributed to this a little more together with Martin Siemert also, who did all the analysis in C. elegans. So we think that at this cluster, Argonaute proteins are phosphorylated. It was shown actually by Josh Mendel's lab in a very nice CRISPR screen, who discovered together with us this phosphorylation cluster, that CK1A1 is the kinase that produces the phosphorylation. We think is a priming kinase that trigger hyperphosphorylation. And then other kinases may come in, and potentially several, and may contribute to hyperphosphorylation and target dissociation. Again, Josh lab published in this screen phosphatase, which is PP6C, and this is in a complex with ANKD 52. And we found there's another component, PP6R1, forming this ternary complex, which is the enzyme that dephosphorylates Argonaute and allows it again to bind mRNAs. So now we can add to the cycle that upon binding and upon specific signals, this kinase is recruited to these complexes here, and then phosphorylate Argonaute. Argonaute can no longer bind to the mRNA and gets released. So this phosphatase complex dephosphorylates Argonaute, and Argonaute may now be able to re-enter the cycle. So we have another, on this cycle, silencing cycle, we have another hyperphosphorylation cycle, where the Argonaute can actually fully lose the microRNA and maybe engage into a reloading is fully unknown. There's no data on that. Probably not, but this is also something we find exciting and we are working on and trying to figure it out biochemically. So where is this phosphorylation event really relevant now? And again, I'm showing a couple of ideas now, which are unpublished. So microRNA-guided gene silencing forms repressive mRNPs. What is a repressive mRNP? The Argonaute protein is recruited. It recruits this TNRC6 protein, and until all these interactions form, the deadenylases are recruited and so on, the mRNA is of course not translated and translation inhibited. So these are mRNPs, and it was shown by Gaspare La Rocca and also later by Amy Buck, and there's a couple of more papers that the Argonaute proteins and microRNAs can be found in high and low molecular weight complexes. And we also tried to recapitulate this, and we looked into, and I cannot go too much into the details, we looked into high and low density cells, resting cells versus proliferating cells, and found that in low density cells, We have a high molecular weight complex where we find Argonaute is this upper band here, and a low molecular weight complex. And when the cell go into resting stage, this high density, we lose this high molecular weight complex and shift it into a low molecular weight complex. And we think that is the Argonaute inactivation. Low molecular weight is just simple Argonaute, and this is the repressive mRNP. And this inactivation goes through the reversible hyper and hypophosphorylation. Interestingly, on a side note, the TNRC6 proteins are degraded in this situation. So we lose these proteins completely, while Argonaute get inactivated but stay there. So they're most likely phosphorylated, which we are analyzing at the moment. So TNRC6 proteins are actively translated, but proteins are immediately degraded. That's what we're working out now. Argo proteins are inactivated through hyperphosphorylation. And that happens when cells go into G1 and probably also to G0. And our model to test is now that there's less gene expression dynamics in these resting cells, most likely in these quiescent G0 cells. So gene silencing pathways are temporarily turned off on a post-translational level. And that may allow to remain in a ready-to-go position, like at a pit stop. So cells are ready, and if the signal comes, TNRC6 proteins can immediately be remade, Argonaute can be dephosphorylated, and silencing dynamics can start again. And this is food for thought. And with that, I would like to end and thank the people in the lab, funding sources, and collaborators. And thank you very much for the opportunity to talk here.
Thank you so much, Gunter. I will start with one question. This protein TNRC6, is it only cell cycle regulated? Is it now what regulates the expression of this, or is it also some stress can regulate, upregulate, downregulate?
So in our case, it's not transcriptionally regulated. So it's ubiquitously made. We find it in all cells. But in our case, it is actually degraded. So the protein is constantly made, and it's degraded through the proteasome and through autophagy. So in this situation, when cells are in this phase, so the protein is made, constantly translated, actively translated, but immediately degraded. So that is kind of allowing a fast response. But we do see these proteins in all cell types and everywhere. But this specific degradation situation, that is in resting cells only.
I see. Great. Thank you for this exciting insights and food for thought. We'll move on. If any other questions arise, please write them in the Q&A. And Gunter will be happy answering them over the chat. So we'll move on with our next speaker, Davor Lesser. So after we know about the mechanistics, let's move on to role of the Argonaute proteins in disease and human disorders. So our next speaker, Davor Lesser, currently heads the Institute of Human Genetics at the University of Regensburg. And previously he was heading the Human Genetics Department at the University of Salzburg. Davor studied medicine at the Medical University of Vienna and did his Ph.D. at the University of Split, focusing on the identification of novel monogenic causes of different syndromes, rare diseases, and he further complemented this research in using different clinical samples in Croatia, Spain, and Germany. And his role is basically addressing the genetic basis of the rare diseases, neurodevelopmental disorders, and mechanisms of aging. And his talk today will be decoding the Argonaute syndromes, lessons from the human disorders. So please take it away. Great to have you here, Davor. Thank you.
Thank you, Neva, for this nice introduction. I hope you can see my screen, and also for the opportunity to share some of our work on the Argonaute syndromes here.
I don't see the full screen. Yes, now.
Okay, no. Okay, so sorry. Okay,
Yes,
so
thank
before I move into the Argonauts, and I also want to thank Gunter for the nice introduction on the basic biology of the Argonauts, so I don't have to talk about this a lot. But I'll just want to give the first slide, the introduction on the basic human genetics, because I'll be talking about these germline mutations. So what are these? These are mutations which basically affect the whole organism. They're found in every cell, and they can be transmitted to the next generation. And unlike them, there are also somatic mutations which are just found in one, which originate in just one cell, and they're found, for example, in cancers. And due to the technological advances in the last years, we were actually able, especially the next generation sequencing technologies, we are also able to identify so-called de novo variants. So these are mutations that are not inherited, but actually occurred spontaneously in the germline. And what we know from the studies performed in the last years is that such de novo mutations, which are not inherited from either parent, are the major cause of neurodevelopmental disorders. So
Doctor,
this is
can I interrupt? Can you please turn on your camera? Thank you.
turn off or turn on?
Turn on. Thank you.
Oh, okay, sorry. Okay. So using these next generation sequencing techniques, especially so-called trio exome sequencing, we identified de novo germline mutations in AGO2 and linked them to a neurodevelopmental disorder. So in our first study, we identified altogether 21 patients, and we identified 12 de novo variants, which were mostly found in L1 or L2, or basically helix 7, or in the PV domain. And interestingly, 5 of these variants were recurrent, meaning they were found in more than one patient, as shown here with the numbers. And then we also identified one patient who harbored large deletion, which involved the first 3 exons of AGO2. So to give you just a brief overview of what these patients have, these are all children. They have all intellectual disability. They have a delay in motor development, also the delay in speech development. And then they have several other neurologic symptoms like muscular weakness, autistic features. Those who learn to walk have also unsteady gait and other gait abnormalities. Almost half of them develop seizures, and then they also have different visual impairments. What is really interesting is that not every patient who has a pathogenic mutation in AGO2 has the same clinical spectrum. So we see really mild to severe clinical spectrum. And interestingly, there is one missense variant which causes mild spectrum, and also the deletion of AGO2, the heterozygous deletion, causes this mild phenotype. So over the years, we have performed several functional analysis, and due to the time constraints, I chose to share with you 3, which I find really key to understand the molecular mechanism of this Argonaute syndrome. And I'm pretty happy that Gunter went talked already about the AGO2 phosphorylation cycle. I mean, we didn't know what each of us will present, but as he has shown, this phosphorylation at the C-terminal cluster is actually associated with the final step of the AGO2 mediated RNA repression, which enhances the release of the target RNA from the RISC complex. So in collaboration with Gunter, we measured phosphorylation of this C-terminal cluster upon immune precipitation of AGO2 from HEX cells, followed by quantitative mass spectrometry. And interestingly, as you can appreciate here, this C-terminal cluster was strongly reduced in all mutants tested except for one variant shown here. And then next, we also performed a QRT-PCR on RNA samples isolated from AGO2 immunoprecipitates. And interestingly, we observed that at all mutants exhibiting the reduced phosphorylation of this cluster also showed an increased association with target RNA. And I'm just showing one here. There is just one variant which behaved in a different manner, and this is this missense variant G733R. And again, this is the missense variant which causes the mildest clinical phenotype. And we performed several experiments testing several of AGO2 functions, and this variant appears to be a complete loss of function. Also here, it's quite clear that she can only poorly bind the mRNA. And this is helpful because it shows us that the other variants actually cause a gain of function, probably due to the impaired release and prolonged AGO2 mRNA interaction. So the next key experiment for understanding was the AGO RNA bind and seek experiment, where together with the group of Ian McRae, and these are also data published late last year, AGO2 and microRNA complexes were incubated with a library of target RNAs containing various seed matches and also mismatches. And what we can see from the results here in the wild type AGO, most RNAs contain a canonical seed. These are shown in different nuances of blue, reflecting high targeting specificity. And then in contrast, when we selected for this analysis 4 missense mutations, most RNAs which are bound by these mutations are lacking the seed matches, and this indicates off-target binding. So these mutants are not simply less active, but they are actually less selective. And we believe that this loss of targeting fidelity is actually the key of this gain of function mechanism that these missense variants show. And the next experiment we did, it was performed together with Hans-Jürgen Krankamp in Hamburg. So to test how these mechanistic differences actually translate in vivo, we analyzed 2 variants on the opposite side of the clinical spectrum. So we used the variant causing the most severe clinical phenotype, which actually causes gain of function, and the G733R, which causes the most mild phenotype. And using the CRISPR-Cas methodology, we generated mice harboring the heterozygous variants. And then we isolated RNA from the mouse cortex. And for the most severe variant, we observed the widespread transcriptome changes. There were more than 200 significantly dysregulated genes. And then strikingly, when we performed the same analysis in the mice harboring the G733R, the volcano plot was essentially flat. So we observed just a very few genes which were differentially expressed. So this indicates that reducing the AGO2 function has surprisingly little impact on the global gene expression, whereas altering AGO2 behavior in terms of gain of function leads to widespread transcriptome dysregulation. And I believe that this data actually really nicely explain why missense variants leading to the gain of function cause a much more severe phenotype than a simple haploinsufficiency. Okay, moving further, as most of you know, AGO2 is not the only Argonaute in humans. And there are 3 others, and actually they share the conserved domain architecture. They all bind microRNA, and they all participate in similar silencing machinery. But they're not identical, and although they are very similar, as you can appreciate from the data shown here from Kotaro Nakanishi's work from 2022, there are several positions that are there are not only unique residues, but there are also some unique domain features as shown here. So the question is, can also variants in this paralogs cause a similar phenotype? And one in the study from 2022, led by Amélie Piton from Strasbourg, has actually shown that similar missense variants spread in different domains of AGO1 also cause a neurodevelopmental disorder. And what was really striking is that several of these missense variants, also the deletion of the phenylalanine at the position 180, are actually identical as those that we identified in AGO2. So not only do the AGO1 missense variants cause a similar clinical spectrum, but we actually believe that it's also the similar underlying molecular mechanism. What I would also like to share with you is a finding from 2015, where they identified 5 children who harbored deletion encompassing AGO1 and AGO3. But if you look closer, AGO4 was deleted actually in 4 of these individuals. And they have actually shown that heterozygous deletion encompassing AGO1 and AGO3, and in this cases also AGO4, causes a neurodevelopmental disorder characterized also by intellectual disability, motor and speech delay, muscular hypotonia, and early feeding difficulties. And on the severity grade, these children are affected in a similar manner as if only AGO2 is deleted. So what do we learn from this? It's in a way this supports that there is a partial redundancy between AGO1, AGO3, and AGO4, in contrast to a more or less unique role of AGO2. So to sum up what we have learned from patients on basic Argonaute biology, I think it's quite clear that AGO2 is the only paralog which is dosage sensitive, and the heterozygous deletion causes mild neurodevelopmental disorders. For AGO1, AGO3, and AGO4, there is a partial redundancy, so single gene loss is probably tolerated, and disease only emerges when at least 2 paralogs are affected. But then in contrast, the missense variants act through a gain-of-function mechanism, either AGO1 or AGO2, and the mutant proteins are not inactive, but they actually become hyperactive and less specific. They bind targets to long and to the wrong RNA, leading to widespread dysregulation of gene expression. And this actually also leads me to 3 key insights. So among Argonaute proteins, AGO2 has a unique and essential catalytic role. AGO1, AGO3, and AGO4 show partial functional overlap. And importantly, the severity of these Argonaute disorders is driven by hyperactivation of microRNA pathway and not its loss. So overall, in Argonaute syndromes, the major problem is not that genes are not silenced, but that there are wrong genes are. Moving forward, we aim to better define the clinical spectrum through natural history studies. At the same time, we are currently linking genotype to mechanism through functional analysis of novel variants, and currently there we have identified together with our collaborators more than 50 AGO2 variants. And importantly, we are modeling these disorders in human neurons using patient-derived iPSCs. And ultimately, we hope that this will not only help us to better understand the Argonaute syndromes, but that we may also learn how we might intervene. And last but not least, I just want to briefly share with you that our functional analysis of Argonaute syndromes utilizing patient-derived cell lines also revealed vulnerabilities in the MyRisk pathway and suggesting a potential novel therapeutic strategy in cancer. The work is currently under review, but it's available on bioarchives. So with this, I want to thank my fantastic team. In blue are highlighted people working on Argonauts, the selected collaborators, families, and individuals with whom we also are in contact to the Facebook group. And of course, thank you for your attention.
Thank you, Davor, very much for this fantastic talk. Great to see already research on RNA can pay off also on the Argonaute. While waiting for people to type in their questions, perhaps I can ask a question. I mean, this was actually both for you and Gunter. Do you know if modified RNAs are more targeted, or is there something, are you also planning to check the level of modification on the RNAs? If there is anything different between AGO binding? Is there anything known on that?
I'm not aware, but Gunter is the expert.
Well, it's not, well, not sure whether I'm an expert. It's not, so of course, anything that changes the duplex formation between the microRNA seed and the mRNA would be relevant. So there could be predicted target sites that actually modified and therefore not bound by a
Right.
and the other way around. MicroRNAs themselves are not modified.
Yeah.
Human microRNAs at least.
Okay, cool. Thank you. One question from Robinson Tribulate. Is there any change of microRNA expression in AGO2 mutant knock-in mice versus control?
So we didn't analyze the expression of the global expression of the microRNAs. What we analyzed was the AGO bound microRNAs, and it's interesting that this severe missense variant preferentially binds isomers.
Okay, thank you. Let's wait some more minutes. You can still type in your questions. So with that, I think slowly we can conclude the session. Let's thank again all our speakers again. And next RNA collaborative seminar will be again in Europe with EMBL and Institute of Technology in Italy on the 22nd of April. So thank you so much and wishing you a great day.